Good afternoon, students. I'm so happy to welcome back our Zoom class, right? Today we are going to discuss very interesting topic, that is multiple alleles. Actually, multiple alleles are nothing but a group of alleles occupied in the locus of homologous chromosome, right, which determine a particular character. That is called a multiple allele. I repeat once again, multiple alleles are nothing but a group of or a series of allelic forms of gene may determine a particular trait in an organism. That is called a multiple allele. So what are the general characteristics of multiple allele, right? So multiple alleles are nothing but, this is a chromosome. I like to draw the structure of chromosome in which you find a number of genes. You find a number of locus where you can find the presence of genes, right? So the place where genes are located, that is called the locus. So these green color indicates locus in which many genes are situated in the center, right? Here, a series of genes, a series of allelic forms of genes are located. So, a group of or a series of genes or allelic forms of genes are occupying the same locus or a locus of the chromosome, that is called a multiple allele in order to determine a particular character, right? So that is called a multiple allele. So for example, I'd like to mention some example, right? In cell sterility, right? Plants, we are going to discuss about the cell sterility by multiple alleles, right? So actually this cell sterility is designated by letter S. So there are a number of genes in the S1 and S2 and S3 and S4 and S5. So these group of or allelic forms or series of allelic forms of genes are occupied in the same homologous, the same locus of the chromosome. Now, if you take a chromosome in which there are many locus, each locus may possess a number of genes. Here, these locus consist of a series of allelic forms. They are S1, S2, S3, S4, S5. It is an example for multiple allele. So, these allelic forms of gene may control cell sterility in plants. So actually, cell sterility is caused by multiple allele, right? I repeat once again, cell sterility. What is cell sterility? Cell sterility is a process in which the pollen grain may not germinate when it is transferred to anther stigma, right? That is called a cell sterility. I am going to show you the structure that you can understand. Now, this is a Gainetian part on which you find it consists of stigma, style, ovary, right? It is a female reproductive part. Here, when stamens are originated at the bottom of ovary, right? Here you can see androecium or a group of stamen, right? Now these stamen may consist of and the lobe. So when and the lobe attains maturity, it will release pollen grain. Actually, the pollen grains, this is nothing but it is a pollen grain. Actually, pollen grains are transferred from anther to stigma by the process that is called pollination. Some pollen grains may not germinate when they are laid down or when they are sitting on the stigmatic region due to self-sterility, right? So there is some pollen grain which are unable to germinate and form useless to form pollen tube. So such a condition is called as a self-sterility. Sterility. Sterility is a process by which pollen grains, they are never germinated on the stigmatic region of veination part, right? That is called the sterility. Here, the sterility is caused by multiple allele. Sterility, it is caused by multiple allele. What are multiple allele? Multiple means many. Allele means an allelic form of a gene. So there is a series of allelic forms of genes are occupied in the locus of chromosome. Why they are occupy the same locus of the chromosome? Because in order to cause self-sterility in plants. So such a condition is called as a self-sterility. It is also known as a self-incompatibility, right? Self-sterility, it is also known as self-incompatibility. Okay. Next we pass on to what are the general characteristics which is of self-sterility or multiple allele. General characters. General characters of multiple allele. My dear student, it is very interesting area. Please understand and follow me. What are the general characteristics? First of all, a series of alleles are situated the same locus of the chromosome. There is a series of alleles are occupied or present on same locus, right? Which are found in the locus of the chromosome of homologous chromosome. That is to underline second one. Mostly these multiple alleles they are responsible for same character. They are responsible for same characters, right? Third one. Actually, the multiple alleles are caused self-sterility. So when we take the two different variety, the one is a wild type, right? The first category is the wild type. Actually, this wild type variety which determines dominant one that controls dominant character. There is a mutant variety. This mutant variety also determines over dominance, right? Or sometimes it is to act as an intermediate. So the periodic ratio of wild type is over dominated by the presence of mutant variety, right? The next one, if wild type variety or the mutant variety which are crossed, right? If wild type variety is crossed with the mutant variety, F1 will be only mutant variety, right? This mutant variety is one, it is going to be expressed in F1 generation, not wild type variety. So the periodic ratio of wild type, it is going to be suppressed by the presence of the expression of the mutant variety. So this is called due to multiple alleles. So this is a general characteristic features of multiple allele. I repeat once again. Simply, multiple alleles are it is a series of allelic forms are present on the locus of a homologous chromosome. Generally, they are responsible for the same character, attaining same character. And there are two varieties of wild type and the mutant variety. And wild type variety is able to determine a dominant character, and the mutant variety of allelic forms of gene that controls over dominant character. So when we class with each other, F1 will be mutant variety, not wild type of variety, right? So this is a general characteristic of multiple allele, right? So now coming to the point, actually these multiple alleles are reported so multiple alleles are responsible for self-sterility. As I said, what is self-sterility? The pollen grains which are unable to germinate on the stigmatic region, right? That is called self-sterility due to presence of some allelic forms of gene, right? Like S1, S2, S3, S4. So as I said, as I explained, this self-sterility is caused by, right? By S1. S1 is one of the allelic forms, and S2 is another allelic forms, and S3, right? Another allelic forms of gene, right? And fourth one, S4, it is also another allelic forms of gene that causes self-sterility. Then fifth one, S5, right? So a series of allelic forms of genes may cause self-sterility. So it was first reported by yeast, right? It has been first reported by yeast in the year of 1925. In case of the self-sterility, it was first reported in case of Nicotiana plant. Nicotiana plant, right? I repeat once again. Self-sterility is caused by a series of allelic forms of gene. They are S1, S2, S3, S4, and S5. It was first reported by yeast in the year of 1925, and this self-sterility was reported in the organism of Nicotiana tabacum, right? It is a tobacco variety, right? In which it has been first reported by yeast in the year of 1925. Actually, he has taken 5 different kinds of allelic forms of gene, right? The first variety is taken. This is a stigmatic region in which there are 2 different organisms, right? They are taken. This is a structure of gynoecium, right? During fertilization, in which you can see the presence of 2 different allelic forms of gene. They are S1 and S2, right? These 2 male gametes are haploid in condition, and when they lay on the surface of stigma, this S1 and S2, they are not germinated. That means there is no formation of pollen tube, right? Because this gynoecium or the stigmatic region or the gynoecium part or the stigma possess S1, S2. Both of them are homozygous condition. Homozygous. Simply the homozygous condition of alleles, if they are found in a plant in which there is no formation, there is no development of pollen tube, hence there is no fusion of male and female gamete. Actually, generally, the male gamete, if it is available in the form of S1, it must be penetrated the stigmatic region and passes through the style and enters into embryo sac and fuses with the female gamete, right? Egg is there inside of the embryo sac. So here, unfortunately, the S1 is one of the male gamete. I mean, one of the pollen grain. It is not germinated because the homozygous condition of alleles or allelic forms of genes are found in gynoecium. So hence it causes a sterility, self-sterility. Here, student, please understand, self-sterility is caused by homozygous condition of alleles. So for example, S1 and S1. Here S1 and S1. So S1 of male and S1 of female gametes are fused. So there is no formation, there is no development of pollen tube. Hence, there is no fusion of male gamete into female gamete. So hence, it is unable to germinate. So it is known as a sterility. Actually, the sterility is caused by homozygous condition of allelic forms of gene. I have shown the picture in which you find the gynoecium part that consists of S1 and S2. So if the same allelic forms are germinated in the form of pollen grain, so they are not germinated. Simply S1 and S2, they are sitting on the stigmatic region and they will not form pollen tube, and there is no fusion of male and female gametes. So hence no fertilization is taking place. So this is called as a self-sterility. Actually, self-sterility is caused by homozygous condition of allelic form, series of allelic forms. On the other hand, I am going to show you another picture that you can understand, right? If you take a heterozygous condition of allele, I am going to show you another picture in which you can understand. My dear student, it is very interesting area, right? Please understand. This is a structure in which it's simple. This is a structure I have taken only a small portion. If you take a gynoecium, now this is a gynoecium pore. Right, you can see now this is a stigma, and sometimes the presence of this is a pollen grains, right? Here, what are they? They are the pollen. So the pollen grains are sitting or laying down on the surface of stigmatic region. I have taken only this portion only, right? Because this gynoecium part also possesses S1 and S2 allelic forms, right? So here instead of S1, S2, S1 pollen grain already we have explained S1 is not germinated, but because of this S1 is a homozygous condition of allele. Instead, heterozygous condition of allele. If instead of S1, S2, if S3 is laid on the surface of stigma, then it penetrate, it gives rise to form pollen tube. This is a pollen tube. So pollen tube is developed from the pollen grain and fuses with the embryonic cell, right? I mean, the fusion of male gamete and the female gamete, and fertilization is taking place. So here, so this kind of condition is prevented self-sterility, right? Students, so hoping that you can understand a homozygous condition, they never form a pollen tube, whereas a heterozygous condition of allele, they form a pollen tube, right? Hence, fertilization is taking place. Next diagram I am going to show you that you can understand. Instead of S2, S3, one more allele forms are involved in fertilization, right? This is a third picture in which you find, as I said, I have explained it. It is a part of gynoecium, right? In which I can take only a small portion of gynoecium, in which there are two different heterozygous condition of allele. It is an allele. Students, I am using repeatedly alleles. It is an allele. The name of the allele is S4. It is able to cause self-sterility. My dear student, S4 is an name. It is an allele. S4. It is an allele. It is able to cause self-sterility. Hence, this is a self-sterility. So this self-sterility is designated by the letter S, S4. Now S4, when S4 is lying on the stigmatic region, it gives rise to form pollen tube, right? And the pollen tube develops, grows, and penetrates the style region and enters the embryo sac and fuses. I mean, releases male gamete in the female gamete. Hence, fertilization is taking place and zygote is developed, right? So, on the other hand, I can say one more allele, that is S3. Because these S4 and S3 are able to germinate because these two different alleles are entirely varied from S1, S2. Hence, they are heterozygous condition. In first case that we discussed, homozygous condition of alleles, they are never germinated. Whereas instead of homozygous, we have taken a heterozygous condition in which S1, S2 are one type and S3, S4 and another type. So we have taken 4 different kinds of allele which are able to cause sterility, in which both of them may be germinated because of the condition is a heterozygous condition. Okay, so when we make a flow chart, when we apply for a Punnett square, we can find how many species, how many progeny are getting sterility and how many progeny are not getting self-sterility plants, right? So, as I said, you know very well, I gave its application, but you can understand the first type, that is, this is a Punnett square. We are solving a number of problems in genetics. Students, it is very easy to understand, right? So, I am going to take only 3 pairs of allele. The first form of allele is maleless, right? Here, it is a male. And then let us take it is a female, right? Stigmatic region, right? So as we designated S1 and S2, right? This is the one form of allele, S1 and S2, and S2 and S3. So like that we have taken S2 and S3. And let us take another allele forms of gametes, right? S3 and S4. Here, let us take S3 and S4. So simply I am going to fuse this male gamete with the female gamete. So please understand, during gamete formation, all the gametes are haploid in condition. Please remember, this is a structure of stigmatic region. I'm talking about this is a structure of stigma. Just imagine, right? It is a structure of stigma, gynoecium. What they possess. I have taken 3 kinds of gynoecium. The first gynoecium consists of S1 and S2. That means S1 and S2 are present in one plant and S2 and S3 present in another plant. S3 and S4 are found in another plant. Right? So we have taken different kinds of plant. Right? So if S1 and S2, yes, as I said, self-sterility is caused by the homozygous condition of allele. Right? Here let us join S1 and S1. S1 and S1. So like that the S2 fuses with the S2. So this homozygous condition of allele causes self-sterility. Here the pollen grains of S1 fuses with the stigmatic region of S1 and S2. They are not germinated. It's simply it causes a self-sterility, right? So there is no formation of pollen tube, hence no fusion of male and female gamete, and no formation of zygote, right? So this is a self-sterility due to self-sterility. This sterility is caused by multiple allele. Students, please remember we have taken a homozygous condition of allele S1 and S2 in male and female. When they are fused to form zygote, zygote is not developed. Zygote is not formed by the fusion of sterility allele, right? S1, S2, S2. So hence it forms a self-sterility, right? Second one, if S1 fuses with another gamete, so we here just imagine, just to separate them, this is a condition of haploid condition of allele. Please separate them. Then you can understand, right? Please separate them. During gamete formation, this is a gamete which are haploid in condition. Please understand which are haploid in condition. So here S2 is there, and the second gamete it is going to fuse with the S2, S1 and the S2, right? So here S2 is there. So hence S2 which becomes sterility. So it is going to disappear. Hence S3. This S3 S3 fuses with twice simply, right? S3 fuses with the S1 and again S3 fuses with the S2. So these S3 pollen grain may be germinated in the stigmatic region of S1 and S2. Please understand, student, due to disappearance of exactly I can say the S2, it is an allele. If it is a line or if it is a region of the stigma of S2, it is not germinated. Instead, S3 is occupied on the S1, S2 stigmatic region in which a pollen tube is developed and fertilization is happening, right? So this is a way by which we can get two different allelic form of organism, right? Here in the case there is no formation of cell sterility. The third one, right? Let us connect third one. If S1 fuses with the here, just I'm going to draw this mention to understand. Here, this S1, S2 are one type of allele, and S3, S4 another series of allele. When they are fused, we can get. So this is a male gamete. As I said, it is a male gamete. All the male gametes, each male gamete fuses once. Each male gamete fuses with this gamete of female. So let us fuse it easily, right? First of all, S3 fuses with it twice, and again S4 fuses with it twice. So you can automatically join them, right? S3 twice, as I said, S3 fuses with the S1 once, and again S3 fuses with the S2, right? And the other again S4 taken. If S4 are taken, it is going to fuse with this female gamete, right? So as I said, S1 and S2. Students, you can understand, right? Instead, I can draw it as it is. You can understand very clearly. Simply S3 of each gamete fuses with the female gamete twice. S3 fuses with the S1. Again, S3 fuses with the S2 once, right? And the male gamete of S4. Let us take another gamete. S4 fuses with the twice. S4 fuses with the again S1, and again this S4 fuses with the S2. So in the case, you can see there is no formation of cell sterility. The sterility is not caused because all the allele, we have four kinds of allele which are heterozygous condition. This heterozygous condition of allele does not cause cell sterility, right? And so all the pollen grains are germinated in the stigmatic region of S1 and S2, right? Okay, let us take another one. Here S1 and I'm talking about the second alleles, right? In that S1 is a separate gamete and S2 is also separate gamete. So here just point in the second gamete of female, here S2 is there and S2. So both the S2 of male and S2 of female, they are not fused because of cell sterility, right? So hence let us take it S1. So S1 which is going to fuse twice. S1 fuses with the S2. Again, S1 fuses with the S3. Students, did you understand here? One of the male gamete, I have taken two male gamete in which one is a cell sterility due to cell sterility condition. Hence, it is not involved in fusion of male and female. Hence, S1 is involved because of its heterozygous condition. And as a result, we got 2 to 4 progeny, right? S1, S2 is one progeny, and S1, S3 is another progeny, right? So we have obtained 2 progeny by the fusion of the first male gamete of the Nicotina plant with the second fusion, right? You can understand. And let us connect to third one, S2 and S3, right? Please repeatedly I'm using S2, S3, S2, S3 because they are the allelic forms of gene, right? So this S2 is there, S2 of male and S2 of female, it is going to be disappear simply due to cell sterility, hence S3. So let us take directly S3 is directly taken. And S3, which is going to fuse with the female gametes of allele, they are S2 and S3, right? So S3 generally taken, S3 is taken twice and fuses with it, right? Here, S2 and S3. Let us take any one, right? S2. Simply understand, you can. I'm going to show you the picture that you can understand. So this is a picture in which you can understand because of all the homozygous condition of alleles are fused. Because we are dealing this one, right? My dear students, we are dealing this one, S2 and S3, right? Here S2 and S2 is disappear, and again S3 and S3 are also disappear because they are the homozygous condition of allele, right? Students, you can understand. Now let's come to the point. These alleles are not germinated because of they are homozygous condition. They are homozygous condition, hence they are not germinated. My dear student, it is very easy to understand. So this S2 is so not fused because of they are homozygous condition. S3, S3 also fused. So both of them are homozygous condition of male and female also homozygous, hence it causes sterility. Hence, there is no development of pollen tube or the stigma region. So let us take a third, next one. S2, S3 of female and S2. So simply you can join. Just compare S3 is a common in both male and female. Hence, both of them are not involved. So let us take S4. S4 fuses with it twice simply, right? You can understand S4 fuses with the S2 and the S4 gametes are fused with the S2, right? You can S3. So this is a gamete that you find. The next category, simply you can understand. I'm going to show the picture. So this is a chart in which you can understand. Here we are discussing only these male gametes of pollen and the female gamete of this. So these conditions are simply self-sterility, and we are discussing this the fourth one, right? So let us take fourth one, S3. So here it is possible to obtain because each gametes are haploid in condition. All the gametes are haploid in condition. It forms. By the students, you can understand. Just separate them simply. This S1 is one of the gamete and S2 is one of the gamete, right? Just separate and you can fuse easily. So we are taking here this S1 which is going to fuse with the here. It is possible to obtain two progeny from each gamete, right? Here male gametes are S1 and S2, the female gametes are S3 and S4. This male gamete, each one fuses twice. So like that, S2 also fuses twice. So let us get S1, S3 one progeny, and S1, S4 one progeny, and S2, S3 one progeny, and S2, S4 another. So it is possible to obtain 4 progeny. All the 4 progeny are heterozygous condition. So they don't have self-sterility, right? Even though we are going to obtain a 4 progeny in the fusion of male S1, S2 with the S3, S4, but there is no possible for self-sterility, right? Because all the gametes, all the alleles are heterozygous in condition, right? And let us take a S2, S3 with S3, S4, and you find here. So S3 is a common, and then S3 of this gamete and S3 of male and female are not involved in such a case. So here S2 and S3, S2, S4, another gamete is formed. So fourth one also, S3, S4 and S3, S4 are homozygous condition, hence all the allelic forms of gametes are not involved in the fusion of male and female. So this is a simply something about of multiple alleles. Simply I can say multiple allele causes self-sterility. There is a self-sterility which is caused by multiple allele of homozygous condition of allele. Simply condition is a homozygous, but nature of allele is a multiple allele. Right, my dear students. So next we pass on to this explanation was first given by yeast, right? In the year of 1955, years of tobacco plant. So next topic is sex determination in plants, right? Sex determination in plants. So here only one example is given, that is maize. So I'm going to show you the picture, a maize plant. If you take a maize plant in which there are two different kinds of inflorescence, one is the male and the other is the female inflorescence. Here the picture that shows male inflorescence, location of male and female inflorescence. Actually, male inflorescence is this is a male plant in which the development of male inflorescence from the apical meristome, shoot apical meristome, that is considered to be a male inflorescence, right? Right. And it is designated by, it is determined by the presence of simply this maleness is determined by the presence of TS plus. I mean, the TS plus is nothing but a tassel seed, which is going, it is an allele forms of gene that determines maleness. The plus indicates dominant character. I mean, the dominant allele TS, tassel, TS means a tassel, right? The tassel seed is determined or determines or controls the tallness, maleness in plant, right? And the female character or the female inflorescence, it is designated by BA, barren plant, right? So this BA plus indicates it's a dominant allele. BA is a dominant allele that controls femaleness or female. So this whether maleness and femaleness. So maleness is determined by TS, dominant allele, and the femaleness is determined by B, small b, small e, right? Plus indicates the dominant character, right? So both of them are determined by, actually generally it is a monocyst plant because both male and female flowers are found on same plant but different branches, right? Different area. So this is a male plant, male flower and female flower, which are found on the same plant but in a different area, right? The male flower is found at the tip or grain form, the terminal condition, whereas the female flower, the female inflorescence is originated from the axis. So axillary, that you can say, female is found in another plant, right? So this is called the monocyst plant, right? So here, which both maleness and the femaleness are determined how it is, right? Let's have a better square. I can take a small. This is a plant. So the phenotypical I can see here small b, small ba, small ba. As I said, the small ba is a recessive condition. It is a recessive condition. Ba, barren. Ba stands for barren plant, right? So this small ba is a recessive condition of allele that makes a flower as a rudimentary. I mean, it is simply the femaleness, it is to be lost by the presence of recessive condition of allele, that is a BA. So these are small BA and small BA and small TS, small TS. As I said, small TS is a recessive condition of allele that's responsible for maleness, right? So due to, if it is found in a dominant character, it is going to determine maleness. If it is found in recessive, it is going to determine femaleness. That means undeveloped staminate form and developed distillate flower, right? So due to presence of TS and BA. So if both alleles are found in a plant, right? Because it's a monoecious plant. Maize is a monoecious plant. So this BA, small BA, and small TS, present. So both of them are found in the plant. That causes simply a double recessiveness. And the lax of silk. Silk is funny. It's a modification of stigmatic region. And the stalk is also on the part. It is a transformed tassel, right? So simply due to the presence of recessive condition of small BA and small BA, small BS, small BS, they are responsible for rudimentary female. Though the flowers are femaleness, but it is unable to fuse with the female gamete, right? So that is a rudimentary property. So on the other hand, I can say one more. I'd like to show you another formation of gametes. Instead of both of them are recessive, one is a dominant and another is a recessiveness, in which we can find this is a condition. My dear students, you can understand here the one allele is dominant and another here a small b, small ba is a allele form, a recessive allele, which is responsible for a determination of the femaleness. Whereas if TS plus, this is a superscript that indicates dominant character. So here TS and TS, both of them are double dominant characters. So due to presence of this TS dominant or TS dominant, and both of them are found in a plant. What is its nature? It is going to be determined by maleness, because this maleness is determined by TS dominant allele and the TS dominant allele. Actually, it is a double alleles are present. There are two alleles are present in determination of maleness. Though femaleness alleles are present, but its character is hidden. But the flower is present, that is a rudimentary condition, right? Female flower is there, but it is in the form of rudimentary. Ovary is not developed well, right? So instead, small BA, let us take small BA dominant one instead of recessive. Here we are going to take dominant one, BA. What is BA? It is an allele for bearing plant, right? That determines femaleness. So this BA plus indicates dominant allele if it is that, and both TS and TS, I mean both alleles are dominant for maleness and the femaleness, right? So both allele forms are dominant. Then what is its nature? It is called simply monoecious. Here both maleness and the femaleness are determined. Because the allele for maleness is a TS and the allele for femaleness is a BA plus. Both alleles are present, right? Hence, the condition is a monoecious. Actually, monoecious is caused by the presence of small BA plus, which are dominant allele. They are responsible for femaleness. And what about maleness? TS plus and TS plus, because TS is plus is a dominant allele. So both the dominant allele for maleness and the femaleness are found in a plant. The plant is determined by monoecious. What is monoecious? Both maleness and the femaleness are found in the same plant and different plants, right? That is called monoecious, rarely dioecious, right? So instead of both maleness and femaleness, right? We have taken heterozygous condition of allele. The one is dominant one. Femaleness is a dominant one. Students, please understand. Femaleness is a dominant and maleness is a recessive. The recessive allele for tarsal and dominant allele for barrier, if they are taken in an organism, what is its nature? It is going to be determined a normal female, because the femaleness is determined by small ba plus, because dominant allele. So dominant allele of ba plus that determines femaleness in males. Instead of ba plus or dominant allele, ts, a tarsal, it is an allele. Tarsal dominant allele, if it is that, it is going to determine maleness. So that is the difference between maleness and the females, right? So this is called simply sex determination in plants. I repeat once again, there are simply two different alleles, dominant allele and recessive. We're talking about dominant allele, BA plus, DA plus. It is an allele that determines femaleness, and as a result, we can get here sex determination in plants. It is determined by the presence of two different alleles, right? Simply you can understand, right? My dear student, simply TS and TS, it is an allelic forms of gene, right? That determines maleness, male flower, right? And here instead of TS, BA and BA plus, that controls, that determines female. We have taken a dominant allele. The alleles are dominant. So if dominant allele TS that determines maleness, and the dominant allele of BA that determines femaleness. So instead dominant allele, when we take a recessiveness, right? TS and TS, it is if it is a recessive condition. So there is a recessive condition, it makes a flower, staminate flower, which is undeveloped one, right? So the male flower in which staminate is gradually staminate. The staminate is gradually eliminated and forms as a rudimentary ovary, rudimentary oval surface, right? So like that, if instead of a dominant allele, we take a recessive condition. Here recessiveness. So small ba with small ba, it also forms. It makes the flower as a pistillate flower is gradually eliminated and forms like a tarsal condition. So hence a rudimentary. So this is a sex determination plants, maleness and the femaleness. Simply maleness is determined by dominant capital TS, small ts plus they are dominant allele that determines maleness, and femaleness it is determined by small ba plus, right? Thank you for joining, my dear students. Anything else you can ask? Students, did you understand first of all? My dear students, right? Dear students, did you understand today? First of all, am I audible? Students?
Yes, sir.
Did you understand today?
Yes, sir.
Okay, self-sterility, where it has been first reported? Name the plant.
Nicotina.
Very good. Could you mention the allelic forms of genes which are responsible for self-sterility?
S1,
S1, S2.
One S2,
S1, S2.
right, very good. My dear students, I have taken a picture. In that you see S1, S2 is a femaleness. Maleness S1, S2. Why they are germinated? Could you answer, please? Okay, no problem. And sex determination plants, where it is first reported, where it was first reported? Name the plant in which sex determination was reported.
maize.
Maize, good, very good. And students, will you mention the allelic forms of gene for maleness?
TS plus.
TS plus. Why it is determined the maleness plant? The plus indicates always.
Dominant character.
Very good. But what about the female? Femaleness is determined by
BA plus. BA plus.
A plus. Very good. So thank you for joining. If there is any clarification asked, or do you want to ask anything? Would you like to get clarification regarding this topic? One of you, could you mention what do you mean by monoecious? What do you understand about monoecious? Students, are you able to follow me?
Male and female flowers are present on the same plant.
Same plant or same flower?
Same male and female flowers are present on the same plant.
Very good, very good. Good. Here I have taken a small BA and small BA and TS plus TS plus. So this form of allele it is going to determine what type of organism, whether male or female. Students, boys.
Female.
Female or male?
Female.
Here I have taken small BA for females and TS plus.
Male.
Maleness, because it is a dominant allele. Understand? So just I have shown the picture that you can understand. Please see, right? So thank you for joining, my dear students. Now I am going to give an assignment. Please take down.